Um, th- Today we're going to have one of our own get to speak to us, and it's Dr. Debbie Carr. Deborah got her undergraduate degree at Colorado, University of Colorado in Boulder. Her master's degree is at UNM in Albuquerque, and then she got her PhD out at TIES. You worked with Todd, didn't you? With Todd Anderson, who's the acting chair and interim director of the Institute for Environmental and Human Health. I asked her what her area of expertise was, and it really, she's very, very broadly trained, has experience in a lot of aspects of different ecological methodology, but I think probably what she's going to talk to us today about is microbial ecosystem ecology. She's been supported really pretty well by a number of grants, especially those dealing with BP and the Event Horizon, or whatever that thing is called, that blew out in the Gulf and had the oil spill. But then there were many, many effects that sometimes were more active than we thought, and sometimes were less. And I can speak to that because I've actually seen those guys down there. I get to go fishing every year for about a weekend, and we go right to the place where they were. But fortunately, none of the hatcheries, I believe, have been affected at all, except those from mollusks. She's going to talk about that research, and she's going to talk about a number of other things that she's done. She told me that the last time that she gave a seminar here, it actually resulted, or the work resulted, in a patent with Carol Heinz on some work, I guess, involving cleaning. That's what I thought, cleaning up oil spills, because that's. So, without further ado, we're glad to have Debbie Carr, and please join me in welcoming her.
Well, thank you. Yeah, I am happy to give this talk because I think a lot of people in the department know who I am, but they have absolutely no idea what I do, besides being married to James Carr. And we have some research projects that are together, but we don't do the same thing. But it's hard to explain that, knowing what he does as a neuroendocrinologist, and me as a soil microbiologist. That's about as far apart as you can get in a biology department. So how in the world do we have projects together? One of the projects I'll talk about today will describe both of our roles in that. But it should be very clear that we are not the same person from this talk.
Thank God.
Okay, so the title of my talk is Microbes at Work. Okay, how microbial communities inform us about ecosystem health. Make it work. Okay. Microorganisms are everywhere. They can live under the most stringent environmental conditions imaginable. If there is any way that they can gain energy from a system, whether through photosynthesis or chemosynthesis, they are able to thrive. Okay? The more we are able to get to extreme environments, the more we are able to find some of these microbial communities going on. And these are not isolated organisms. These are thriving communities interacting. They are completely intertwined with all of the living systems on Earth, from the human gut to deep sea hydrothermal vents to Antarctica, and in some of the most contaminated situations you might imagine. So, ecosystem ecology of microbial communities. Microbial ecosystem ecologists spend a lot of time communing with microbes, okay? Understanding the structure and function of the communities and the microbial role in processing nutrients through the ecosystem. They're at the very base of all of our ecosystems. They are responsible for decomposing things, for recycling the nutrients and soil structure, and basically everything that drives primary production of an ecosystem. So most of our ecosystem services begin down at the microbial level. For instance, the nitrogen cycle is very important, but it takes an entire community of microorganisms to complete that nitrogen cycle. Each different population completes one aspect of the chemical transformation of that nutrient. Understanding how the chemicals move through our ecosystems requires understanding the microbial soil ecosystem from the community point of view. There's a whole lot of microbes out there. There's a whole lot of diversity. They're everywhere. The soil and sediment environments offer an amazing amount of niches to provide that diversity. There's incredible gradients in temperature, in soil moisture, and the oxygen available, and carbon and the nutrients available. And there are microbial populations that can take advantage of every one of those situations. They look for chances to outcompete another population. They're very good at it. Microbes are very easy at evolving, adapting. They can evolve. They can pick up traits that their neighbor has just by bumping into them, like our new cell phones. And one of the most frustrating parts when you go out to sample these systems is you can bring in a soil or sediment sample and put it in some water, mix it up, look at it under a microscope and see a ton of different microorganisms. We cannot cultivate hardly any of them at all. Less than 10% of them are able to be cultivated and put into cultures. If we cannot put them in cultures, it's very difficult to learn anything about an individual population. Part of this frustration is, well, part of it's very real to me, as I'll show you in one example, but a lot of this frustration is what has driven the microbial ecologists to utilize, try to horn in on the molecular genetic techniques with deep sequencing, metagenomics, things like that, that are probably not, they may not be essential tools for the ecology, but they're the best we have to be able to look at the differences between things we can't culture. Okay? With sequencing, we can tell that things are different. In microbial ecology, we don't necessarily care what their names are, but we want to know that there are different populations. So it's with all that, with the lack of being able to culture and study these things in the lab in a controlled situation, we know very little about what makes them tick, what makes their community structure the way they are, how they carry out functions, who carries out, who provides cofactors for one population, and what is the stability of their community, and how much redundancy and resiliency is there in that community. If we could bring them all into the lab and culture them and repeat them and do exact replicates like you can with cell culture and things like that, it would be a lot easier. But if it was that easy, we wouldn't have the complexity that we know that exists out there. So a large part of the challenge here is changing from a pure culture type model and an approach into one that includes the community diversity and all of those complex factors, including the abiotic factors, that shape the microbial community associations. That complexity drives the biodiversity, okay? If you're a microbe, there's a lot of jobs to do. You can always be employed. There's always something for you to do, some way for you to survive. So why do we care, okay? The soils and the organisms that live in the soils are crucial to the biodiversity of the organisms that live above, live in the ground above them, walk on the ground, crawl on the ground. Without that soil structure and the nutrients in that soil, the other biodiversity that we more often encounter doesn't exist. They provide essential ecosystem services. No one else does nearly the job of recycling and decomposing organic matter back down to its elemental parts. And those nutrients are necessary for the primary production that drives the system. It's easy to imagine then, if you think of it from a microbial ecosystem base, of the entire ecosystem collapsing from the base down, the base up. Okay? So today I just want to give a brief overview of some of the microbial environments that I have done studies in. Some of them are natural, and some of them have been man-made environments. Lechuguilla Cave and freshwater sediments are natural environments. I'm going to discuss those first. And then some wastewater effluents, and the rest of the studies are all related to hydrocarbon spills, petroleum spills, and associated type products. Okay, so Lechuguilla microbes. This Lechuguilla is a fairly newly discovered cavern in the Carlsbad, New Mexico area. It was first, the entrance hole was first excavated in 1986. Now that seems like a long time ago to some of you guys. I was in grad school at the time, and coincidentally, there were some friends that were extreme cavers in the department, and they were involved in this first opening and some of the first people involved in the cave. Now this, as soon as they got in there, they went back out, and they started, and they're grad students, you know, they said, this is a pristine environment. This is a very nutrient-limited environment. There's no sun. What do we know about this system? Absolutely nothing. What are the chances we're going to ruin it before we know anything about it? And right from the very beginning, they were able to work with the park system to limit how many people, who could go in, where they camped when they were in there, what they had to take out, which was enough to drive me not to go down there, because you took out all your waste. Okay, all your waste products. But at the same time, they came and asked me if I would be interested in helping look for microorganisms and potentially identifying some microorganisms in a cave system that is brand new open. Okay, so it's obviously a chance you don't turn down. Some of the microbial habitats that we looked at there, and again, I say we, that's the royal we, they went down in there and collected the samples, were pools. Some of these were puddles, very shallow depressions. Some of them were lakes 28 meters deep. All right. We looked at sulfur, manganese deposits, iron deposits, some of the corrosion residues from these iron deposits. And we also looked at the human camping areas, the places where the humans had been camping, okay? They took sterile swab samples for fungal studies and cultured those out and isolated 37 different species of fungi and in 13 different genera. The bacterial samples were taken at air-water interface, at sediment-water interface, and from cores in some of the formations. And immediately, some of these samples were fixed in a glutaraldehyde solution to fix them so they could be looked at for SEM studies. And then the other samples were brought out, and they gave them to me, and I started trying to culture them. So we plated these out on oligotrophic media. Okay, this is not an environment that has a lot of nutrients, so a nutrient-rich media is not going to tell you anything useful. Used anaerobic media, used chemoautotrophic media, and after a lot of work, a lot of plates, a lot of replicates, we were able to isolate 8 unique colonies. And these were identified then, these colonies were identified by fatty acid analyses as belonging to 7 different aerobic bacteria and 1 anaerobic bacteria. And that anaerobic one is potentially a facultative aerobe. So it's primarily anaerobic, but it can exist in limited oxygen environments. These bacteria, we could not specify, we could identify by fatty acid analysis, which does not give you down to genus species level. It gives you family relationships. It was very frustrating. We still didn't know, are these bacteria indigenous to this cave system? Were they brought in? Are they so ubiquitous in nature that they just are there, coming in through air currents and things like that? It was very frustrating. All that work was great, publications out of it, and it was still frustrating, okay? And I've been frustrated with that ever since. So, above ground, about at the same time, I was also working in a stream system, a first-order stream, in New Mexico, and looking at the nutrient cycling going on in the streams, the microbially mediated nutrient cycling. Okay? This system was very unique, and what it consisted of was we had 3 first-order streams. The first-order stream is a spring-fed stream. It has no branches, nothing else contributing to the stream reach except for the spring at the headwater. And these streams were of different sizes. One of them, at the top one here, had a beaver pond on it. And this was our third one, which was a very narrow little stream running through this grassy meadow, and it was about 18 inches wide. It was free-flowing, there were no obstructions in it. And then the middle stream was a similar stream to this, but we actually created there was a flat area in there, and we created a beaver pond and introduced beavers into it with different success stories depending on who you speak with. That's a whole other topic, though. And what I was finding in I was sampling the interstitial waters of these systems, and I was finding a lot of anaerobic spaces, a lot of ammonia, a lot of methane in these systems. Now, it's not hard to think of methane or anaerobic soils in a system, an area with a beaver pond, okay? It's covered with water, it's marshy, smelly soil and everything. That's anaerobic. But in a system like this, right through there, why would you expect to see any anaerobic sediments. And so we started looking harder. And I was able to do a comparative study at 3 different LTER sites, the long-term ecological research sites, in the Consul Prairie in Kansas, Coweta, and H.J. Andrews. And what we did there was sample the interstitial waters, look for methane, and do nitrogen anaerobic, nitrogen and ammonia fluxes in the systems through these chambers that I had to make through PVC pipes. They were not real sophisticated, but they did work. So what we found is there was an interstitial methane in the pore waters in all of these LTER sites, just as there had been in the New Mexico streams. Okay, the H.J. Anderson site had methane even in it. It was low, even though these streams are very cobbly. Okay, you don't have a lot of fine sediments in them. You found the most in the Consul Prairie streams, which are a lot more characterized by silty and clay soils. And Coweta was low, but we did find interstitial methane there. The important thing here was that we looked at methane oxidation rates, the rate at which the bacteria were utilizing that methane as a carbon source to grow on. And you can see that in all of these sites, in the pools and the riffles, methane oxidation, the utilization of methane as a carbon source, was very active. Now, this wasn't done, these samples were taken one week in one place, 1 week in another place, 1 week in another place, okay? In the summertime. It was one single point in time. The original study that drove this, and you don't have to see all of this table here, but these were the first 3 streams, the Beaver Pond stream, and this was pretty characteristic every year that we did it. Around 30% of the samples that we were seeing were anaerobic. Again, that's not that unusual when you think of a beaver pond area, but when you look at that small, free-flowing stream, a lot of primary production going on there, you still had 25 to 46% of the samples collected in the summertime were anaerobic. Okay, so microsites within aerobic sediments are very productive parts of the interstitial waters, and very, very crucial for cycling your methane, your carbon, and your nitrogen. There were a lot of anaerobic sites. They were not, they weren't evenly spaced. You couldn't say that, okay, there's a dead log, partially buried log, and you were going to find an anaerobic site there. They were a lot more heterogeneously spaced than that, non-predictable. They varied. This one place, I had standpipes so I could sample the same sites every time. One time it might be anaerobic, and another time it might not be. Okay, so these sites vary temporally and spatially along these streams. Dissolved organic carbon, methane, ammonium nitrate, and dissolved organic nitrogen, all of these nutrients are enriched in interstitial waters compared to the surface waters. That's not that surprising. And then ammonia, DOC, and methane are very much enriched in the anaerobic waters over the aerobic interstitial waters. Methane is a major component of the dissolved organic carbon. Okay, we saw that from the methane oxidation studies. And then the dissolved nitrogen phosphate ratios in these streams in the anaerobic zones is increased because ammonia is so so enriched in these areas. So these anaerobic areas are hotspots of nutrient availability. Okay, so those are some natural systems. All right, and these were a long time ago, but they really started me thinking about these microsites, these areas we don't spend a lot of time thinking about and are not necessarily characteristic of the larger site. So I started thinking, well, how do we know a system is stressed? Okay, what happens when we know a system, or we would expect a system to be stressed, and because we see pollution there? Okay, how do microbial communities react to that? And this, at that time, was just kind of a vague feeling and thought, okay? But how do we know if a system is stressed? Well, most of the time it's because we work on a certain species and we go into a system and say, this is not doing too well here. Okay, our indicator species is not thriving. Or the entire primary production of a system is down. We can also see it because the soil structure is in decline. And we can tell soil structure is in decline if we've got compacted soils. We've lost root zones in the soil. Or if there's a lot of erosion going on in a soil. That soil is not a fertile soil. It's not maintaining its soil structure. And it has an impact on the primary production and the diversity of well. So what do we do about it if we have a system that's in stress? Well, we try to understand what occurs naturally, write a grant, and keep studying it. What can we do? There's a potential to remediate some of these systems. And when it's human-caused, a pollutant put into a system, we can try to remediate it. When I was in New Mexico, we started getting asked a lot to help remediate some sites around the state. And bioremediation is using the microorganisms that are present in the soil to chew up and degrade the pollutant compound. Okay? You will try to understand the system and encourage them to do this as fast as possible. So some of the sites that some of the compounds that I was involved in remediating were diesel sites, leaking underground storage tanks, benzene, TCE, crude oil. And the first crude oil site I worked on was Navajo Refinery, and it was involved in it was a designated Superfund site. And they had been procrastinating. They did not want to spend the money that was required for them to start cleaning up their site. And they were being warned that if they did not submit a plan for remediating that site, the EPA would come in and do it for them and charge them for it. So they came to us and asked if we could help design a plan. They sent a plane up to Albuquerque, and we got off the plane, and I'm positive they were in shock because this was a bunch of good old boys in the oil industry in the early 90s. And getting off the plane was a gorgeous southern girl from Georgia, and me with a 3 or 4 month old baby in tow. Okay, and we were going to go tell these boys how to do their business. The plan we came up with was approved by the Superfund committee for an initial remediation attempt. Okay, And so that was, they did not come to us because they were trying to be environmentally savvy. They came to us hoping for us to find a way for them to do this on the cheap, okay? Microbes don't get paid a lot per hour. So we were happy. I mean, we were happy to do this. Our motivations were different from theirs, but it all met in the middle there. Pharmaceutical degradation I've looked at, and also product testing by Shell Corporation for drilling fluids. I've been involved in it. So what are some of the things you try to do when you go in to bioremediate something? Well, you really have to understand the chemical structure, the compound structure of the contaminant you're interested in. You have to know those functional groups that are involved, where they are, well, how strong the bonds are, what kind of energy is going to be required to break those bonds, what nutrients might be provided if those bonds are broken. You need to understand what the degradation products are. When it starts to be broken down, what products are going to be forming intermediate, because oftentimes those degradation products or daughter products are more toxic and more labile in the system than the original parent contaminant. You don't want to make a situation worse, so you have to understand these things. You also have to understand the ecology of the system. You have to understand the nutrients in the system. What is limiting the system? What's limiting the microbes from doing what they can do? Or what could you create? What could you cause to be limiting in order to force those microbes to go scavenge that nutrient from the compound that you want them to start breaking down? And you can use these tools to carry out remediation. The disciplines involved in it are ecosystem ecology, which is what I was trained in at the time. And I did a lot of those projects with only that background. You start working on some of these projects and things like TCE. Get a little scary when you start thinking of a lot of these daughter products that are created. And you quickly realize that some remediation things maybe ought not to be done in the environment, and maybe a reactor site is better. And this entire aspect of it is where people are interested in cloning in super microorganisms and things like that, to use in reactors to break down a lot of these really nasty compounds. So I was realizing I know absolutely nothing about molecular biology, and first chance I got, I went over and spent 14 years in a molecular biology lab, learning a lot of molecular biology, doing a lot of Western blots, a lot of cloning, a lot of enzyme kinetics, a lot of sequencing. If you guys don't know, in the modern world, this is an old sequencing gel. We don't have to do those too often anymore because DNA sequencing has become automated and so cheap it's a real waste of your time to do it this way unless you have a very specific need. Another aspect that I realized I really knew very little about was chemistry and the analytical chemistry involved. If you're going to tell somebody you can clean something up, you ought to be able to show them that you've done it. Okay? Measure the compounds that have disappeared and what's being created. And so I spent another 4 years in an analytical chemistry lab learning cool things like HPLC, GC, GCMS, a whole slew of analytical chemistry. Machines do not scare me. The most exciting aspect of this whole thing is the ability to use next generation DNA sequencing. And it's relatively inexpensive to be able to send it off to a company to do it as well. So, the next 4 cases I'm going to talk about are some of these contaminant cases. Pharmaceuticals in our wastewater, okay? This has been a topic that's been in the news a lot. And our wastewater systems have not been designed to be able to remove these pharmaceuticals and personal care products. Our wastewater systems are designed to remove nutrients, nitrogen, phosphorus, and carbon. If we don't want to be drinking this stuff, our water treatment systems are going to become a lot more sophisticated and a lot more expensive. With that comes an idea that people want to be able to use wastewater effluent as irrigation. Their thinking is it's less expensive than drinking water, which will increase the freshwater drinking supply. Nitrogen and phosphorus that are in that effluent are actually fertilizers for their crops, the grass. They're not subject to water restriction on irrigation. If you're a golf course, that's a nice advantage. And importantly, anything that you do apply to these areas of grass will help recharge aquifers. But what about the pharmaceuticals, personal care products that are in that effluent? What happens when you use that for irrigation? Fortunately, Lubbock has an extremely good test site for this because the city of Lubbock has applied land application, their effluents, to land application for about 70 years. These areas, these center pivots between 50th Street and 19th Street, right outside of the Loop, all belong to the wastewater treatment plant, and those center pivots only put out effluent water. Okay? So if you want to go see if there's contaminants building up in the soils from these pharmaceuticals, what a better place. Okay? The nice thing about center pivots is, and the corners of these areas, is you can use the same soil types for your controls that have never been exposed to these. So in the big study, I was looking at a variety of estrogen compounds, some antibiotics, and some personal care products. Today I'm just going to show you some results from estrone and triclosan. Triclosan is an antimicrobial. The difference between an antimicrobial is a synthetic antibiotic. An antibiotic, per se, is something that's made within an organism. Okay. Other than that, they both kill what they are applied to. The thing when you're talking about personal care products and effluent is we want to study them individually, but they don't occur individually. They occur in that effluent or applied on that ground as mixtures. So, but mixtures are really difficult to study. So this was a very simple mixture study, just 2 equal amounts of estrogen and triclosan. And we're looking for a variety of, well, 3 simple explanations. Is they both degraded at the same rate. One, estrogen slows and triclosan remains the same, or they both slow down. 4, so if here is the estrogen degrading organisms are resistant to the triclosan effects, and there's no limiting nutrients in the system. In this one, the triclosan is toxic to the estrogen degrading organisms. And in this one, they're both degrading but slow, so there may be a competition for resources there. It was a very simple study, 10 grams of soil, spiked with 25 parts per million estrone and triclosan. These are very high concentrations. I did it aerobically and anaerobically, and then we extracted them and ran them with reverse phase HPLC. Okay? So individually, the estrone and the triclosan both degrade. The half-lives range from a half day to about 7 days. For estrone and for triclosan, about a week to about, what is that, about 15 days, something like that. Putting them in a mixture, looking at the estrone, it still degrades, but it's very much slower. The triclosan is still degrading, but it's way slower. Okay, so just like most good science, it didn't fit with any of your predictions because the estrogen didn't slow as much as the triclosan did. Okay? There's a variety of possibilities for that. It just wasn't something we expected. So you can see here, these are just the differences in half-lives. From a half day to 16 in the best case for estrone, and 3 days to 44 days in the worst case for estrone degradation. Triclosan goes from best case 6 days to 79 days, or 80 days best case, down to over a year for a half-life degradation of triclosan. Now, the interesting thing on triclosan, if you notice from the structure, it does look an awful lot like thyroid hormone. So there may, there's reasons to be interested in this entire process. And if you're interested in that, James Carr has a lot more knowledge and data on that at that. So the conclusions, anyway, from this study are that in a mixture, both of those compounds degrade slower. Okay? Estrogen degraded even in the presence of an antimicrobial, but triclosan, the antimicrobial, degraded even worse. There may be a definite competition of resources going on here since they were both slowed so dramatically. So that is a question that deserves a whole lot more study. The next study was a shell drilling fluid evaluation. Now, when you're an oil company and you want to extract oil, you dig a hole and you apply a drilling fluid to cool that drill bit, but also that drilling fluid brings the rock pieces out of the borehole that you're drilling up to the surface where you deposit it into a pit and eventually spread it over the land and land farm it to dispose of it. Okay? Conventionally, they have used diesel as a drilling fluid. Less toxic alternative has been to use low toxicity mineral oil for their drilling fluid. So Shell is interested in developing a new drilling fluid. They call it a gas to liquid drilling fluid. It's synthesized from gas, methane. And the thinking for them was because it's a cleaner burning fuel, less aromatics, sulfur dioxide, particulate matter, things like that when it burns, that it may be a better alternative as a drilling fluid as well. I have absolutely no idea at this point whether it's better or worse for the soil or the things that grow in the soil. So they asked some people out at Ty and myself to take a look at this. We looked at 2 different soils, one from Texas, one from Nebraska. I'm just going to show you the Nebraska stuff today. The treatments, we did, the first GTL1 was a C10 to C22 compound, and the GTL2 was a C11 to C24. Okay, so there were different lengths in the branches of the hydrocarbons in these. Unless you're a petroleum engineer, you probably don't care. We tested it with low toxicity mineral oil and also diesel. We also included a vehicle control because in order to mix these things into the soils, we diluted them quite a bit. We diluted them with acetone, so we tested the acetone as well as soil that was not treated. As part of this study, there were invertebrate toxicity studies, seed germination assays, and microbial toxicity from a CO2 generation. The parts I was most involved in was degradation of the hydrocarbons and microbial diversity.